Hello, my dear Butcho, and welcome back. I know, exams are just around the corner and the tension is building up. But don't you worry. Today we're going to tackle one of the most important and honestly, one of the most fascinating chapters, Principles of Inheritance and Variation. Let's make sure your concepts are absolutely crystal clear so you can ace this topic. Full marks, Bucka!
So let me ask you a simple question. Have you ever just looked in the mirror and thought, I have my mother's eyes, but I definitely have my father's nose? It's not some kind of magic, my friends. It's purest science. It's genetics. And by the end of our little chat today, you'll know exactly how all of this works.
Okay, so here's our game plan. Super simple. We'll say hello to genetics, meet the hero of our story, the great Gregor Mendel, understand his two main laws, see some really cool exceptions to the rules, and then a super fast recap to get you completely exam ready. Simple, right? Let's jump right in. So first things first, what even is genetics? Well, think of it as the study of how traits, characteristics, are passed down from one generation to the next. It's the science that basically explains why you look a little bit like your mom, a little bit like your dad, and a little bit like your grandparents. And the most important word here is heredity. Imagine it's like a secret family recipe. Your parents pass this recipe down to you, and it has all the instructions for making, well, you. Your height, your hair color, even the shape of your nose. That whole process of passing down the recipe, that's heredity.
For thousands of years, heredity was a huge mystery. Nobody could figure it out. But then, a humble monk, working in a quiet little monastery garden with nothing but simple pea plants, he cracked the code. He solved the whole puzzle.
And this is the man Gregor Johann Mendel. We call him the father of genetics for a very good reason. His work was just revolutionary. For the first time ever, he used maths and statistics to explain biology. He brought logic and numbers to this great mystery of life. Now, Mendel's real genius was his choice of the Humble Pea Plant. It had 7 very clear, very distinct, contrasting traits. For example, have a look here. A plant was either tall or dwarf, no in between. The seed was either round or wrinkled. This black and white clarity made his experiments so incredibly easy to track and analyze. So smart. Okay, let's get into his first big experiment. It's called a monohybrid cross. Now, mono means one, right? So he decided to study just one character at a time. For instance, just the height of the plant. But wait, before we go on, we need to know this very important word, allele. So gene is the instruction for a trait, like height. Alleles are simply the different versions of that instruction. So for the height gene, you have a tall allele, which we show with a big T, and you have a dwarf allele, which we show with a small t. Easy peasy. So here's exactly what Mendel did. He took a pure tall plant, that's big T, and crossed it with a pure dwarf plant, small t. And what he found in that first generation, the F1, was just amazing. All the baby plants were tall. It was as if the dwarf trait had just vanished. Then he let these F1 plants self-pollinate to create the second generation, the F2. Now, to predict what would happen in the F2 generation, we use this super handy tool. It's called a Punnett Square. Just think of it like a simple grid, okay? We put the alleles from one parent on top and the alleles from the other parent on the side. Then we just fill in the boxes. And boom, we can see all the possible genetic combinations for the offspring. It's a simple way to see probability in action. And when Mendel actually counted all the F2 plants, he found something incredible. For every 3 tall plants, there was 1 dwarf plant. The dwarf trait that had disappeared in the F1 generation, it was back. This consistent 3:1 ratio of what you can see, the phenotype, this was a major clue.
But if we look at the genes themselves, the genotype, the story is a little different. The Punnett Square shows us that the actual ratio of the gene combinations is 1 pure tall TT, 2 hybrid tall TT, and 1 pure dwarf TT. So that's a ratio of 1:2:1. Remember both of these ratios, Bacho. They are very important for your exam.
So this one simple experiment gave us 2 of Mendel's biggest, most important laws. First, the law of dominance. This just says that in a hybrid, one allele, the dominant one, our capital T, is the boss. It will hide the effect of the other, recessive allele, T. And second, the law of segregation. This says that the 2 alleles for any trait separate from each other when gametes, like sperm and eggs, are formed. These are absolute key concepts for your exam, so make sure you understand them really, really well.
Now, Mendel's laws are the foundation, the base of everything. But genetics is full of surprises. Sometimes the rules get a bit more interesting. Let's see what happens when it's not so simple as just tall versus dwarf.
So we know about simple dominance, where one allele is the clear winner. But sometimes you get incomplete dominance. Here, neither allele is the boss, so you get a blend in the middle. Think of a red flower and a white flower making a beautiful pink flower. And then there's codominance, where both alleles are expressed equally. They're like partners that both show up to the party loud and clear.
Let's take a look at a really beautiful example of codominance. Here are the parents. On one side, we have a pure red-coated cow, and on the other side, a pure white-coated cow. Now, what do you think happens when they have a calf? And here it is. Their calf is not pink, it's not light red, it's something called roan. And if you look closely, you can see it has patches of red hair and patches of white hair. Both the red allele and the white allele are showing up completely. They are co-dominant. They're coexisting. How cool is that?
Okay, ready to level up? Mendel didn't stop with just one trait. He thought, what happens if we track two traits at the same time? This is what we call a dihybrid cross. Di means two. So here, Mendel crossed a plant that had round and yellow seeds with a plant that had wrinkled and green seeds. Now, as you'd expect, in the F1 generation, only the dominant traits showed up, so all the seeds were round and yellow. But when he crossed this F1 generation, just look at this massive Punnett Square for the F2 generation. You can see all the crazy different combinations that are now possible. And when Mendel sat down and counted all of these F2 seeds, he found this beautiful, consistent, almost magical ratio, 9:3:3:1. That's 9 round and yellow, 3 round and green, 3 wrinkled in yellow, and just 1 lonely wrinkled in green. This proved that the gene for seed shape and the gene for seed color are inherited independently. They don't affect each other. This is his second great law, the law of independent assortment. Phew, I know. That was a lot to take in, but you all have been doing so well. Now for the final and most important part. Let's do a super-duper quick revision of all the key fundas that will absolutely guarantee you get full marks on this topic. Okay, students, focus here. Take a mental screenshot of this slide, or a real one, I don't mind. These are the 6 things you absolutely must remember. The 3 laws, dominance, segregation, and independent assortment, and the 3 magic ratios, 3:1, 1:2:1, and the big one, 9:3:3:1. Just memorize this, and I promise you, you are completely sorted. You have all done a fantastic job today. You have mastered the basics of Mendelian genetics. But I'll leave you with a little thought to chew on. Mendel's law of independent assortment. It works perfectly when genes are on different chromosomes. But what happens when 2 genes are really close together on the same chromosome? Do they still go their separate ways? Or do they travel together as a team? Well, that's a story for another time. Keep up the great work, study hard, and all the very, very best for your exams.